Okay, so as I said, we may produce Biofloc within 30 days, no problem. But according to this, because we are facing the EMS problem, so we need to reduce the production or the formation of Biofloc as soon as possible. All right, so this is what we want. So up to 4 ml per liter Biofloc. So this is harvesting period of shrimp. So this is some background of fundamental study, how we calculate, on how we isolate the bioflocculent producing bacteria. We work together with the main producing shrimp in Malaysia, known as Blue Archipelago or Aisha, here in Terengganu, not too far from our campus, university. All right. Firstly, we isolate from their farm, and then taking all the sample and so on. When we do have, we try to screen for the most potential bioflocculent producing bacteria, and from that study, we found that there are we isolate bacteria, fungi, and microalgae. All right. So, but among these three, we finally managed to get the bacteria is the most potential candidate to be used as inoculum as compared to fungi and also microalgae because of their reproductive or their multiplication. All right. So this is the first case study that we implement or try the first trial on the using the rapid BFT or rapid Biofloc TM. So we are working with one of our friends or industrial partner in Perak. Right. So as I said, this is same problem regarding disease, environmental effects. All right. So we try to use this rapid Biofloc, and then this is what we're proposing. So before we grow them out in the grow out pond, we ask them to prepare a treatment pond. All right, and then we are using this nursery pond. And up to 1 gram or at least 1 gram, then later on we transfer to the grow out pond. All right, so the optimum water quality value are, all right, so we managed to get the temperature, pH, salinity, dissolved oxygen within the optimum value. All right, so I would say I would like to conclude this by having the optimum conditions for this shrimp. Right, so in terms of nutrient, ammonia and nitrate, so we managed to get less than what we are targeting at the moment. All right, where we managed to get lower than 0.2 and also 2 ppm for the whole treatment. All right, I would like to share as well the production. So as we can see that here, so production in terms of grow out one using rapid Biofloc, and also we're using the control. So control meaning that they are using the only feed or pellets as a monitoring or as the feeding. All right, and then you can see that the survival rate is much more higher in a grow out using rapid Biofloc. All right, and the production as well. All right, so the impact is improving the shrimp health and productivity. So we can basically.
Dr. Nur Azman, yeah, the time is 3 minutes left. I think it is maybe you can pass on your.
All right. I will finish within the time frame. Okay, thank you, Inita. All right, so basically there is no indication of disease occurring. All right, so maybe I can, okay, almost finished. Okay, so the second case study will be on the impact of rapid Biofloc on the development on the microbacterium or udang galah here in Malaysia, zooic culture. So as you know that development of larvae culture, there is 11 step of development of microbacterium before they turn to post-larvae stage. So the treatment will be the control, feeding just fed with only Artemia. The second one fed with Biofloc only, and the third one combination of Biofloc and Artemia. So what we found that basically feeding with microbiome Rosenberg with Artemia and Biofloc have increased the turning over into the post larvae up to 27 days or 29 days. All right, then this is the other study that on the grow outs of giant freshwater prawn. So basically, and also this is we try to see the development of giant freshwater. Instead, normally in the pond, we without Biofloc, we as a control, so that we basically can produce not less than 5 months. But in our case, we may produce or can harvest within 3 months. So we believe that this Biofloc do something on the growth performance of udang galah. So I just skip on the mud crab, and this is basically on the development of Biofloc on the jet perch production. All right, so this is how we collaborate with industry on producing premium size of jet perch. And the outcome, novel application, because we never tried this on the jet perch before. And as a conclusion, this rapid formation of Biofloc by injection has increased the production and also shortened the production periods of any aquaculture production. And the most important thing, we try to eliminate or protect our aquatic ecosystem with using the beneficial microbe within the Biofloc. I think that is from me. I hope that I give within this short of period of time. Thank you, Dr. Yunita, for giving me time. Thank you very much.
Thank you very much, Dr. Nur Azman. It was a very interesting and informative lecture. So we have understand the Biofloc from theory to application. And now I would like to invite our second speaker, Professor Karin Pittman. Okay, yeah. The microphone.
There we go. I hope you can hear me now. Can you see me? Oh,
yes.
Can you see me?
Yes, we can see
okay,
you.
I do not see myself, so I'm not quite sure how it looks. I'm extremely pleased to join you, and I'm extremely honored to see how well Dr. Yunita has done after she's been at our department. And I'm also very, very happy that I am not the only one who cannot adjust their background here. So I join other speakers who are unable to make the virtual background. I'm also very thankful to University of Brawijaya to allow me the opportunity to speak with you. And I'm also extremely grateful to Dr. Nur Azman, who has made a very good introduction to the approach that we will be doing here. I want to share my screen here, which is this one. And let's see. Okay. I have only a few slides, well, a few relatively speaking, but I've also prepared, come on. I've also prepared a PDF of a larger set of slides, which are talking about the details behind what I will be saying. And my point here is that we have to advance aquaculture beyond agriculture, because our planet itself is poorly named. It is not planet Earth, it is planet ocean. And therein comes a big deal about how we think about ourselves and what is like us. So the content of my talk will be about how aquaculture is not agriculture underwater, a bit about the risk management, fish and shrimp-based health indicators, the external immune system, which dovetails very nicely with the topics that Dr. Nur Azman was talking about, and the environmental health and aquatic animal health, and some conclusions. And at the end, also on the PDF of these slides, are some recommended resources, which are all free, highly updated, and very aimed at sustainable development of the aquaculture industry. So you may or may not have heard about Norwegian salmon, and that has been the backbone of the aquaculture industry in Norway. So in terms of national production, Indonesia produces about $18 billion worth of aquaculture products, seafood, every year. Norway produces a little bit over $6 billion. A lot of it is based on the salmonids. So even though a lot of my slides may have pictures of a salmon, the principles that I'm going on are basic principles that apply to all fish and most shrimp here. So one of the things that we have done in Norway, it's a very small country, relatively speaking, only 5 million people, and the governance has been very intense. And this is like the roles of institutions governing aquaculture production, both the ways that it is supported, how it is regulated, how things are registered, how the health is managed, and every single piece of medicine is registered in a local database. So a number of things are very regulated within Norway, and the risk assessments for the environment and the bottom environment for the wild fish and so on, and the European food safety authorities, and then there's the scientific support and so on, and the National Veterinary Institute and so on. But key in this for Norway, as for Indonesia, is that the management of the industry is actually the management of people, and the product-based indicators and management has often been a little bit off the side. So what we've been doing and looking at this as a basic industry, after 50 years of intensive aquaculture, we know the fish is an animal better than we ever did from millennia of fishing. And that means that now we're understanding the whole life cycle and what impacts on it. And that makes it a very different beast. This slide here is a summary. In the PDF of this talk, there are details about this, but the aquaculture is definitely not agriculture underwater. The first thing is that land animals, from the time sperm meets egg to when it's slaughtered, it can spend up to 50 to 70% of its entire lifespan in a protected, stable environment. If anything goes wrong with that stable environment, you have mortalities, you have deformities, you have other problems. Fish spend 0 time in a protected, stable environment, and they are genetically suited to adapt to the variable environment. So they have 0 time in a protected, stable situation. As well, evolutionarily, they have been gifted a third round of whole genome duplication, which means they have not 1 copy of genes on their chromosome, not 2 copies, not 4 copies like we have and like all animals on land have, the second round of whole genome duplication. Fish went through a third round of whole genome duplication, and that means 2 times 4 is 8 copies of the genes on the genomes, many of them to do with the housekeeping of developing in a variable environment and tackling the entire aquatics. The third big difference is that on land, and for us, if we scratch like this, it makes very little difference. We have dry skin on the outside, and it doesn't hurt. However, if you scratch on a fish, that slimy layer is living cells, and scratching them off makes a big difference. It's part of the immune system, the innate immune system, and that is a very special and important biological layer that we have ignored and been overlooked, that it is the key. It is what links what Dr. Nur Azman was talking about the environmental quality, the water quality, the microbiome, and how the fish or the shrimp is experiencing these environments. That slimy layer is exactly where we need to go for how to maintain health, because this is where we have to consider that when we are regulating aquaculture, We cannot regulate it like it is an agricultural animal. We cannot beat the ectoparasites off it. We cannot dry it out. We cannot brush it, because these living cells are part of the first barrier against disease and against mortality. So when we are trying to change the focus, we have to recognize that the biggest risk in the entire aquaculture industry, whether it's fish or with shrimp, disease, and the biological challenges of disease. That is across the board and throughout the industry. And that means that if we change our focus from battling disease to maintaining health, we get a completely different way of looking at this. For the salmon industry, we have been unfortunately looking at the clinical diseases, and it is an open, dirty secret that between 20 to 25% of all the salmon produced are lost between the hatchery stage and harvesting. 25%, 1 in 4. And we're not quite sure exactly how. Well, a lot of it is because the effect of having an outside immune system is really important. How to regulate it is also important for being able to say that you have a sustainable production. And many of the qualities for sustainability are based on exactly what Dr. Noor Azman was talking about, the water quality and the microbiology and whether there are pathogens and so on. But as anybody knows who's been trying to look at fish behavior, it's happy, it's sick, it's parasitized, and then suddenly it's dead. So on the behavior part, it is really, really difficult to determine that a fish is a little bit sick until the real disease has taken hold and their appetites drop and they start to die. And so what we need are directly auditable indicators where a standard includes an associated measurement quality. And this little summary I have put up here is part of the overview of what the Scottish industry has looked at in terms of indicators of fish welfare. It is linked at the back of this, and it is free for downloading. It has looked at 5 different environmental equality and sustainability certification programs. This is ASC and a number of others. And those that indicate physical health on the fish and the shrimp are almost zero. There are very few product-based health indicators. Now, when I say this is often controversial because even with PCR, you can go down to the gene level. But now, remember, there are 8 copies of the genome on the chromosomes, and many of them have other things to do, and many of them as well are involved in housekeeping. But also, we all, every single one of us, has bacteria and viruses and parasites that are on us, but we consider ourselves healthy. So the presence of a pathogen is not the same as the presence of a disease. We can easily carry a few parasites. It won't hurt us, and they are not intending to hurt the host organism. So this is where we come into what our work has been focusing on, the slimy layer on the outside of fish and on shrimp gills. It is an ancient protection against waterborne pathogens, and it is hair thin. It's about 0.07 millimeters thin. And it is that slime. Those are the living cells and the living interactivity here. So what we have there is that fish and all aquatic animals have had for 500 million years a conserved external immune system that is the mucosal epithelia. And what we're showing here is a salmon, because that's what we've been working on. And the blue coloring here on the small inserts for the skin and the gills and the guts is the blue coloring that comes from staining mucus cells, which produce the slimy layer, with periodic acid shift alcium blue, a very old and stable way of staining for glycoproteins that are in the mucosal matrix here. And it is the same on all these structures. Even though the skin is on the outside, we call it the shield, and the guts on the inside. It is the inside of the it is the outside of the inside lining there that is the mucosal epithelium, and it is also the outside of the gills as well. So we call the skin the shield. The gills are the sentinel, the first watcher of the environment. And the guts, of course, they are swallowing the water. They are swallowing all the pathogens in this. That the mucus and the slime is important for survival has been clear for a very long time. These living cells are the first protection. It is more important to keep a layer of living cells reproducing than it is to keep the skin intact. So cutting the skin, in this case, there's a reference for this one here. Cutting the skin gives a higher mortality, a lower mortality than actually drying off the mucus. And this is to an exposure to Vibrio bacteria, which is extremely common in the aquatic environment. So when we're considering this mucus layer, it is a living, learning, interactive user interface. And it happens all the time. It's conserved from the time it is in an egg stage. It is evolving its response to the environment throughout the life cycle. And it also learns, and it has to learn very quickly when it is changing environment from, say, the broodstock to the hatchery, and the hatchery to the nursery, and from the nursery to the grow-out farms. So the mucus is part of the innate immune system, which is more important than the acquired immune system. As you all know, the organisms in water have to reach a point where they are immune competent, and many of them are not. This immune system also recognizes pathogens that we haven't even thought about. It recognizes that, for example, when the grow-out cage nets have lots of fouling on it, these organisms on the fouling are, for example, hydroids that have a neurotoxin in them, and the gills themselves react to this. But we have not considered that a pathogen, and we're not often worrying about cleaning the nets. We're thinking clean is good. Clean is particles, and those particles may have many, many sources. Yes?
Yes, you have 5 minutes left.
Very good.
Okay.
What we have then is this user interface. And so what we want to do is keep the organisms from being exposed to the bacteria without a good protection. This also means that since water is pathogen soup and it's always there, what we want to do is keep the innate immune system active. And this also includes this unilateral eye stalk ablation of the broodstock shrimp. It increases disease vulnerability. It gives an open access to all the pathogens that are in the water. And there is good reason to rethink again how we are transporting this, how we are exposing and stressing the shrimp. That this is carried on through the life cycle is very clear when you look at how the water quality impacts, and I've circled the production here, which is the broodstock, the juvenile production, the disease management, and of course how you treat the diseases with the mechanisms we have. So the environment is extremely important. what we have done with our mucosal mapping is we have made a standard that allows us to look at how the skin and the gills are changing in their responses and which environments are the most difficult to handle. This means that we are able to look at the skin and the gills that are built up the same way with a standard. The green is where the medium of all the clinically healthy fish have been for the skin and for the gills. The gills have fewer protective cells. They get there when they have to be. And for us, it is in the sea-based, the open water, where the gills are starting to really recruit and respond to the pathogens in the water. So this means that the health of the environment is the health of the aquatic organisms. And this is why water quality is extremely important, but also the impact of the pathogens there. The organisms themselves that we are producing are responding to the known and the unknown pathogens. And they are the ones we have to listen to, looking at their innate immune system. There are a wide variety of environments in Indonesia, and some of them have been in not good quality. This affects the ability to produce in any array. So what we have here in this short talk is the aquatic environment cannot be managed like agriculture. Aquatic animals have an external immune system, and keeping this intact is very important. We need shrimp and fish-based auditable indicators of health. One of them is how this mucosal immune system is functioning. We are working on shrimp now to allow us to get a standard for seeing how new health relationships exist and how they interact with, say, Biofloc. And the impact is, in fact, we have one planet, one ocean, and one way of having the health. So the biodiversity is extremely important. It is one planet, one ocean, one health. We wish that our stocks will be as plentiful as yours, Bineka Tunggal Ika, because we need this biodiversity in all things, not just the one. And for the resources that are free and available online for download, they are the welfare indicators. This is an enormously well thought through resource. There's a poster of the welfare indicators. Even though it's mostly for salmon, the same things apply to all the other fish. There is how to do what the robust fish is actually like. That is on the website for the little company we have called QuantiDoc, which is quantification and documentation. There is the sustainability and aquaculture information for Norway. It's always updated, so it's a live, usable resource. Farmed fish welfare practices and the best aquaculture practice standards available for the BAP certification program. So you can start to look at what fish or shrimp-based health indicators there are. Thank you very much.
Thank you very much, Professor Karin Pittman. It was a very interesting lecture because you also provide the data from Indonesia, the aquaculture from Indonesia. Thank you. And now we are going to have a discussion with questions from the viewers. And then for questions, you may fill the Google form. You can write the questions and to whom you want to ask the questions. And we will read your questions and the speakers will answer your questions afterwards. We have questions now already. Number one from Dr. Julie to Dr. Nur Azman. Yeah. The question is, what kind of microorganisms or bacteria are present in rapid biofloc? And how do you verify that the biofloc actually contains the bacteria coming from rapid biofloc? And how much we need to inoculate a shrimp pond? Maybe the first question first to Dr. Nur Azman. Please, Dr. Nur Azman, to answer the question.
Okay, thank you. Thank you very much for the question, Dr. Yuni. So, Dr. Yuni asking about the nitrate catastrophe for the part of Vietnam. How could we overcome the problem? Okay, so we should start or we should think about the first problem in the aquaculture. Basically, where is the nitrate or ammonia come from? So basically, in general, there are two ways. Basically, for the first one will be on in the feed that we given to the fish or shrimp or whatever. All right. So and basically, those fat will be used by the shrimp and later on to get the biomass of the shrimp and so on. And then also the other thing is regarding the nitrate concentration will increase when the feces accumulated in the pond. All right. So when we go back to the nitrogen cycle, basically nitrogen will be nitrifying. Firstly, will be converted into ammonia, and ammonia will be nitrifying. Will be nitrified to nitrate NO2, and then later on and also NO3 nitrate. All right, and later on denitrifying to the atmosphere as a nitrogen gases. So this cycle need oxygen, high level of oxygen. So it will basically be flush back if we don't have the oxygen. So what we can do is instead of using the biofloc as a alternative solution, it's not just only single solution. We can increase the aeration system in the aquaculture system. Let's say we may optimize the dissolved oxygen up to more than 4 or 5 or somehow we can reach up to 6 ppm. So to maximize the oxygen concentration or the dissolved oxygen in the water. So this is the basic thing. All right. So otherwise we need to know the feeding regime. All right, because somehow shrimp or fish are not using whole feeding. So somehow it will turn to the uneaten feed. So I do hope that it could answer the first question, Dr. Yuni. All right.
Yes. Thank
you very much. All right. So as regard to the question by Dr. Julie Ekasari, I do hope this is my good opportunity to meet virtually Dr. Julie Ekasari. So because I quite frequently cite her paper or her works regarding before this. So today I very happy to see her virtually. So Dr. Julie, so I think I have answer some of the question in the chat box, but in order to share with others, so the rapid biofloc is consists of gram-positive bacteria. I hold this now. So can you see this?
Yes.
All right. So this is basically 500 ml of biofloculum producing bacteria. So we use this as inoculation, inoculate. So normally we suggest to the farmers to use this inoculum up to 1 liter for 1 hectare water. So 1 liter hectare of a farm. So we just only use this one at once. All right, once the bioflocs are already formed, so we don't have to put any more injections. So because we know that this is just only can be used as a starter culture, and after that we need to monitor and know how to calculate the C to N ratio, CN ratio 15, and so on. All right. And regarding the second question, how do we verify? So basically, we have done a few trial before, both in hatchery or in a lab scale, and also field trial. We manage. we managed to isolate and identify the most dominant species that we found in the second date of culture and the next following date of culture are the bacteria that consist in this water, in this inoculum, sorry. All right, so basically it's dominated by bacillus. So as we know, as aquaculturists, bacillus is a probiotic bacteria. Right, and how much product that we need to inoculate? So basically, as I said just now, in principle, we just use a very small amount of inoculum. So we propose to inject 1 liter of inoculum. So this is 500 of rapid biofloc into 1,000 tons of culture water. All right, so it's a big amount of, and then we add up with CF ratio and provide sufficient aeration and only applicable in closed ecosystem. Don't use in free flow water. Otherwise, this inoculum will flowing through the river water. And last but not least, how to maintain the stable biofloc during grow out in the pond. As I said, the most important thing is that we maintain the carbon to nitrogen ratio, and please make sure the dissolved oxygen in the water, because this is a live bacteria. They need oxygen to respirate. So that instead of providing them dissolved oxygen, sufficient dissolved oxygen, our culture organism like shrimp, fish, and others need also dissolved oxygen. Otherwise, they will compete each other. So make sure the rule of thumb is that we just develop this biofloc at sufficient only. I do hope that I answered all the questions. Thank you very much, Dr. Yunita.
Thank you very much, Dr. Nur Azman. It was a complete answer for the questions. And now the question for Professor Karim Tikman. I read for you, maybe. The first question is how to manage wastewater generated from the end of the implementation of aquaculture activities. Yeah. And then maybe the previous slide. Okay. And then the question from Dr. Ani. Disease in shrimp or fish is a secondary infection. How to get rid of the disease during the culture period? And then from Dr. Julie, if the mucus layer plays an important role in fish immunity, is there any nutritional approach to enhance the quality and quantity of mucus layer in fish in terms of protection against pathogen infection? Yeah.
Oh, thank you very much for the questions. These are actually right at the heart of what we're trying to do, and if we look at the disease as a secondary infection, as the first questioner has stated, how to get rid of the disease during the culture period is that's one of the problems that we've been having. Trying to treat the disease also introduces greater stressors and another environmental challenge to the organisms that we're trying to produce, the shrimp or the fish. So when, for example, we try to treat for an ectoparasite, we are in fact drying out, scraping off, or impacting on this mucosal layer. And the gills themselves are often very much impacted, and people don't look at that. That is where the respiration occurs, that is where metabolism goes, and that is where an awful lot of the fighting along the environment can occur. So what you want to do is try to follow the fish as they are healthy, and keep fish or shrimp as they are healthy, and monitor when the gills are changing their thickness and their shape. And one of the ways that we've been doing this, and it's extremely sensitive, is looking at the differences between, they always say hyperplasia and hypertrophy of the mucus cells and the epithelial cells on the gills. And we've developed a way in which this is standardized and not not requiring a perfect orientation of the section that you're looking at. The scientific results are under the website that hosts the robust fish at the end. This mucus layer is extremely important, and I love the question: is there any nutritional approach to enhance the quality and quantity? And in fact, that's exactly how we got started, because the answer is a clear yes. And one of the reasons why we got started was that in about 2009 and 10, in the salmon industry, people were saying certain feeds gave a thicker mucus layer or more mucus, and that gave fewer parasites, less treatment of disease, and healthier fish. But every time they tried to measure the mucus, I mean, they could see it, they could feel it, but they couldn't figure out how to measure the mucus in a way that was understandable and sensitive enough. So we had been measuring all kinds of wonderful things for a very long time. And we developed a method to measure the mucus cells that was independent of orientation, that gave us universally applicable units of measurement, and that was completely unbiased. So for that, since then, we've been able to work with commercial companies, both in terms of breeding, prophylaxis, and especially feeds, to find out which are working best for which stages and so on. I wish I could tell you there was a magic ingredient, but basically we do see that those ingredients that most mimic what is natural in the wild environment are those which are improving the health, or at least not inducing a kind of low-grade inflammation or the trend towards enteritis. When you consider the organism as a whole, you have to think, okay, yes, the skin is the shield. It can probably rebuild the mucosal epithelium fairly quickly. And if it does not, your treatment has not been working well. The gills are the first responders. If there's something in the water that it is infected, that is not appreciated by the fish or the shrimp, you will see it on the gills, on the expansion of the mucus and on the expansion of the epithelial cells. In the gut, on the other hand, this is where it gets interesting. What is in the gut is, much like humans, related to the microbiome, not just in the water, but also in the feeds that you give it. And this is where you have the interaction between the mucus layer, the content in the feeds, and how the organism is activating or deactivating its immune response to the diet. We all have heard about irritable bowel disease. If you make that link from irritable bowel disease and gut problems and think that all animals have more or less the same way of tackling what goes into their stomachs, we know that we would feel very bad if our guts were inflamed. The same applies to fish and shrimp, but our ability to read it is much more impaired because we can't ask them, and they have very few behavioral changes other than the reduction in diet. So the enhancement of the quality and the quantity of the mucus layer, it is extremely important, and the diet here, I think, has been under-evaluated, both in terms of broodstock, the broodstock production quality, and in the finishing diets here. So I would think we are what we eat. If we quality, we become quality and strong, and it is much, much cheaper to keep healthy than it is to fight disease. Thanks.
Thank you, Prof. Karim, for the complete answer. So I think we have, okay, we still have 2 questions for Dr. Nur Azman. How to manage the wastewater generated from the end of implementation in aquaculture activities in the Biofloc system? And does it not pollute the environment? And the fifth one, we have what's the preventive measure to control the high content of ammonia? And then what the recommended farming techniques in the Biofloc system? And what is the desired optimum level of oxygen for the flock system in the indoor tank?
Okay, thank you again, Dr. Yuni, and also for the question, Dr. Ahmad Murjani, sorry. So how to manage the wastewater generated from the end of. So basically, as I said in earlier of my presentation, showing you that the biofloc is contain beneficial microbes. All right, but anyhow, since even though this is a beneficial microbes, it's still living things that need to be considered. All right, so from our experience, when we applied the rapid biofloc in a closed culture system, basically, the slush that normally occurs during without the biofloc are not happening in during when we applied the biofloc. So somehow we are hoping that the biofloc can reduce the impact of the environment, basically. All right. Anyhow, let's say during the draining or the harvesting period, so we need to drain or discharge the water. All right. What we can do is collecting the biofloc itself. What we have done is we collect the biofloc as a secondary production or a secondary product that we may use, and we already in our investigation actually we use that biofloc and use it as a formulated diets, all right, for juvenile. So we found that the protein in the protein content in the bioflocs that we harvest is basically as high as 20%. So I think that is good enough for as if we can
give me the attention present link here. Thanks.
Sorry, I cannot hear.
Dr. Nur Azman, you may continue your answer.
Okay. All right,
yeah,
thank you.
Sorry, it was a disruption. Dr. Nur Azman, your microphone is muted. We cannot hear you.
All right. Can you hear me?
Yes.
All right.
Clearly.
So in conclusion, instead of being discharged the good biofloc we produce from aquaculture pond, we can collect them and make it an alternative feed as a alternative feed, okay, for shrimp or others. All right. So the second question will be, what is that, Dr. Yuni? It's regarding the dissolved oxygen content, right?
Yeah, we will have the questions on the screen.
All right. The preventive measures to control the high content of ammonia. All right. So I think that partly I have answered it on the first question where we need to know how much feeding that we need to give to the shrimp or the fish. All right, the feeding regime. The other thing is to maintain regularly the optimum value of ammonia in the system. I do hope that maybe other colleagues can answer me if in addition towards that. And the recommended farming techniques in biofloc system if I can go historically. Biofloc system was firstly introduced or found by our professor Yoram Avni Malaysia in Belize. Okay, they use this biofloc for rearing the tilapia or the orchomis. So we have already investigate this the usage of biofloc to others. So in our case, we have tried to monodon, waname, jet perch, skyla or crab, and also the microvacuum rosamajai, or in here we call it as a Malaysian giant freshwater prawn. All right, so there is not only for that, but we can do for other aquaculture organism. So last question will be on the required level of DO will be the nice one is more than 5 ppm. I think that's answer. I hope that answer all the question. Thank you very much.
Okay, thank you very much, Dr. Nur Azman Hassan and Prof. Karen Pittman. So maybe I can conclude that the successful of aquaculture industry is depend on the good management of people and also using the management of environmental management with the application of Biofloc, one of them, and also to manage the fish or shrimp health management by observing the mucus outside the body. So because the mucus could be the representing the health of inside organ of shrimp and fish, which is expressed outside the body. So, ladies and gentlemen, thank you for viewing the first session. I would like to thank again for all the speakers for the informative and interesting talk, and also the audience for your active participation. Hopefully, the presentation will be beneficial for everybody. Thank you for attention. Good afternoon and good morning. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi
Waalaikumsalam warahmatullahi wabarakatuh.
Thank you very much, Dr. Yunita, Dr. Nur Azman, Professor Karin. Thank you. And let's proceed to the next session. And for everyone who needs the attendance presence, please kindly wait. We will share in a moment. Dr. Ating Yunirti, time is yours.
Thank you. Very good afternoon, ladies and gentlemen. Welcome to the second sessions of International Webinar of Aquaculture, Environmentally Friendly Technology. I'm Ating Yuniarti. It is my pleasure to be a moderator for these sessions. First of all, may I briefly introduce to you our distinguished speakers, Dr. Julie Ekasari from Bogor Agriculture Institute University, and Dr. Muhammad Fajar from Universitas Brawijaya. Dr. Julie Ekasari got her bachelor degree from Bogor Agriculture Institute University. She granted for her master and a PhD degree from Ghent University. She got PhD degree in Bioscience Engineering in 2014. Dr. Ekasari granted many research grants, and in this year, she is doing research concerning the role of microbial management in Biofloc system in controlling fibrosis in shrimp productions. She also have collaboration with Altec Indonesia and House Wellness Food to conduct the research on feed additive and probiotic supplementations and shrimp culture. She has recently published her articles about Biofloc-based co-culture system of Nile tilapia and red claw crayfish with different CN ratio. Our second speaker is Dr. Fajar. He finished her bachelor degree from Bogor Agriculture Institute University. He got master degree from Wageningen University, the Netherlands, and he granted for doctoral degree in the field of biology from University of Erlangen in 2011. She was also awarded from various research grants and still working for current project concerning effect of squid ink extract on growth hormone of white shrimp infected by white phasis disease. He recently published his article entitled of Curative Impacts of Squid Ink Extract on Hemocyte, digestive enzyme, and CPA gene expressions of Paname against white phasis syndrome. In this session, each speaker will talk for about 20 minutes in parallel. After that, we will have a discussion for about 20 minutes. Please kindly write your questions in the Google form with its link is shared in the chat. Dr. Ekasari will talk in the first turn with the title of Biofloc Technology as an Integral Approach to Sustainable Aquaculture. And Dr. Fajar will talk for the next turn with the title of Squid Waste Product for Increasing Shrimp Immunity. Please give a warm welcome to both speakers. Dr. Ekasari, please, time is yours.
Thank you very much, Dr. Dr. Opting. Okay. Okay. First of all, okay, please allow me to share my presentation. Wait. Okay. Okay, so first of all, I would like to thank the organizing committee from Brawijaya University for inviting me in this special event to share what we have been doing in the development of Biofloc technology. In particular, Biofloc technology as an integral approach to sustainable aquaculture. So as it was introduced by Dr. Ating, my name is Julie Ekasari. I'm from the Department of Aquaculture, Faculty of Fisheries and Marine Sciences, IPB University. So I know that based on what we heard earlier from Dr. Azam, we know some information about Biofloc technology. So maybe I will share again a bit. Maybe there will be some similar information, but I hope it will strengthen your knowledge in Biofloc technology. So as an introduction, we know that in this June, the FAO has released the latest report on the production of fisheries and aquaculture production. But today I would like to draw your attention to this graph showing the proportion between fed and non-fed aquaculture production. As you can see from this graph that the production of fed aquaculture species has been remarkably increasing as compared to the non-fed aquaculture species, which also grow, but at a much slower rate. And what could be the cause? In our opinion, this significant increase in fed aquaculture species production is mostly because of the increase of aquaculture intensification, where high density of aquaculture species in per unit of space or per unit of volume is significantly higher than that of extensive culture. Now, the characteristic of intensive aquaculture is the dependency on external feed, because natural feed that is available naturally in the culture medium is no longer sufficient to supply nutrients that is required by the cultured organism to survive and to grow. And in this regard, of course, in the future, we can expect that the aquaculture will require more external feed and accordingly the raw materials to produce the feed. And what is also important is that the use of external feed also bring about the potential increase of waste production. Why is this happening? So I just want to introduce you, or maybe to remind you to how the waste is produced in aquaculture system. So in intensive aquaculture system, we mostly add external feed in order for the fish or for the shrimp to grow and survive, and that after a period of time we can harvest the biomass. So this is how we want it to be. But in reality, in most aquaculture system, there are also nutrient waste produced from the feeding activities. We know that when we add the feed into the culture system, there will be some feed remain unconsumed, which can contribute to the accumulation of solid waste. And the consumed feed will be digested, and the indigestible material will be ingested as fecal waste, which also contribute to the accumulation of solid waste. As for the digested feed will be absorbed and utilized as for various metabolic processes, so that the fish or the shrimp can grow and can survive in the aquaculture environment. However, the metabolic processes will also produce some byproducts that are excreted to the gills or urine, and contribute to the accumulation of dissolved waste in the water, together with the mineralization products from the solid waste or other organic matter in the water from the activity of the decomposer. Now, the problem is this nutrient waste can directly or indirectly becomes toxic compounds that could hamper the growth or even the survival of the cultured organism. So we need to take this waste out from the culture water. And the most common way, and it's because the simplest way, is by changing the water. Of course, this is the easiest way. So by changing the water, and if there is no further treatment, we actually transfer the waste, either solid waste or dissolved waste, into the environment. And as Dr. Azam mentioned previously, this will create another problem in the surrounding environment, including eutrophication, deterioration of water quality in the natural waters, and so on. And of course, this also contributes to the bad image of aquaculture products that could reduce the marketability of aquaculture products. And what is also important is that we have to remember that the waste in intensive culture was actually coming from the feed, and we actually buy this feed. And the feed is not cheap. This is feed is expensive, and it comprises about 50 to 80% of the production cost. So we need to find a way to make feed efficient, to make a feed utilization efficient, and also at the same time reduce the waste introduced to the environment. So how can we do that? One of the strategies is to apply Biofloc technology. Dr. Azam has also previously described what is Biofloc technology, but maybe I will give some more information, or maybe the same information. So the principle of Biofloc technology is actually to recycle the nutrients within an aquaculture system. So the dissolved waste and also the solid waste is converted into microbial biomass by the manipulation or modification of the C/N ratio in the water through the addition of external carbon source or modifying the feed, so that the nutrient waste can be minimized, and the microbial biomass that is formed in the system can form aggregates that we call bioflocs that can be utilized back by the shrimp or the fish, or can be harvested and use it as feed raw materials. So that in an ideal biofloc system, we can have an efficient use of nutrient utilization, feed nutrient utilization, and also we can reduce the waste discharged into the environment. So in the application and in many research concerning biofloc technology, we observed that indeed in biofloc system, we can minimize the waste, especially nitrogenous waste, which is the most common waste in nutrient waste in aquaculture system. And because we don't have to do water exchange anymore, we can have more efficient use of water. So the water utilization per kilogram of fish or per ton of fish produced can be reduced. And what is also important, we can improve the feed utilization. And with the development of biofloc research and application, we also observe that there are some research including in our university showing that the application of this system can improve the immunity of the fish or the shrimp, as well as in increasing the reproductive performance. Although the latter needs more research to confirm, but we have some strong evidence about the effect of biofloc system on the fish reproductive performance. So that is the illustration of biofloc. So what is biofloc in reality? So this is just an example. In front of our house, we installed a fish tank with the size of 5 tons. Because we are now working from home, so we installed the tank at home. And if we take some water sample from this tank using IMHOF cone, this is a cone, and we will see that some suspended solid will settle down at the bottom of the cone. And if we take the samples under the microscope, we will see some microbial aggregation like this, and where it consists not only various types, various species of bacteria with different function, but also some microalgae species. Sometimes we can also find some yeast or fungi, and we can also find some suspended particles which are trapped in the biofloc matrices. And we can also find some microbial grazers such as protozoa like this, and annelids, and ciliates, and rotifers, which are associated in bioflocs microcommunity, which overall these bioflocs can be consumed back by the fish or by the shrimp. So that is an introduction of biofloc technology. So what we have been doing so far in the university in terms of biofloc technology application and development, we have done many research, including to test the applicability of this technology in various aquaculture species, such as gourami, eels, Asian catfish, including live food such as moina and daphnia, and seracs, freshwater lobster. And we also did some experiment in biofloc utilization as a feed raw material. So we included the biofloc biomass in the feed. We did for shrimp and Asian catfish. And we also did some basic research in the mode of actions, including for example, for biofloc formation, what are the mechanism? How can they make bioflocs, and then what are the factors affecting the microbial composition and the biofloc nutritional quality, as well as the nitrogen removal rate, as well as the mode of action of biofloc, different biofloc characteristics on the aquaculture species response, including in growth, health, and reproduction. But what I would like to share today is mostly about the application at commercial scale. But before I present the result of our studies, I would like to again remind you all that the success of biofloc technology application is determined at least by 2 aspects. The first is the successfulness of nutrient conversion from nutrient waste into biofloc biomass. And the second is the utilization and the consumption and utilization of nutrient coming from bioflocs by the cultured organism. because what usually happen, especially in commercial scale, is that we can convert most of the nutrient waste into bioflocs, but the conversion rate into bioflocs is much higher than the consumption rate by the cultured organism. So what usually happen in biofloc system is the accumulation of biofloc biomass. So this is the main topics of our research in at commercial scale experiment. So the main question is how to control biofloc quantity and functionality so that biofloc can be utilized optimally by the cultured organism. So to address that question, first is how to control biofloc quantity. So to address this problem, we tried an experiment by installing a settling tank that was connected to fish tanks, which the main objective was to make the settling process of biofloc density in a fish tank becomes easier, so that we can take biofloc density biomass from the settling tank without interrupting the mixing and the aeration process in the fish tanks. Because yes, we can do some biomass collection from fish tank directly, but the problem is we have to during the settling process we have to turn off the aeration and the mixing, and this can cause some reduction in oxygen level, and also the fish movement can still disrupt the settling process of bioflocs. So based on this experiment, this is the result of the experiment. Although the overall production performance of both biofloc system was considerably good, however, we can see that in this table that the addition of settling tank can improve the growth and the production performance of the red tilapia in biofloc system. And what also important is that we can reduce the feed conversion ratio up to down to 1.18 as compared to the FCR in the conventional biofloc system with about 1.2. And based on this experiment, we observed that the addition of settling tank could reduce 50% of biofloc density. However, the water quality was quite similar, indicating that that we can still improve or enhance the density of the fish so that we can find the maximum capacity of this settling tank. In terms of biofloc quality, we have done some research at the laboratory scale, and based on that research, we found that a biofloc with a particle size of more than 100 micron is actually a good protein quality, because this bioflocs has essential amino acid index of more than 0.8. So AAI is an index that indicate the quality of a protein source according to the essential amino acids composition. And our previous study also showed that the utilization level and biofloc functionality in terms of nutrition significantly affected by particle size and microbial composition. So based on this information in the laboratory, we tried to do another experiment with giant prawn or udang galah. Here we did the experiment using 2 different types of microalgae, which is also chosen based on our previous experiment showing that microalgae addition could increase the biofloc particle size and nutritional quality. So in this experiment, we use 2 different species of microalgae, chlorella and Ankistrodesmus. And we did the experiment outdoor using 1 cubic meter tank So this is mainly for giant prawn nursery culture. So indeed, the result of the study showed that the addition of microalgae, especially Ankistrodesmus, as you can see here in the pictures, that the microalgae addition significantly increased the particle size of bioflocs. And if you see these are the graphs showing the particle size distribution. The green graph, this is showing the treatment with Ankistrodesmus addition. However,
Dr. Julie, sorry, you only have 5 minutes left.
Okay. I think this will be my 2 last slides. Okay. So in terms of fatty acid profile, we can see in this graph that the addition of microalgae was actually modified the composition of short chain fatty acid. Why we use short chain fatty acid is mostly because we know that short chain fatty acid can be used as a biomarker to see or to determine the relation between trophic levels in an aquatic ecosystem. So based on this short chain fatty acid profile, we can calculate the correlation between the fatty acids profile in the bioflocs and in the giant prawn. And we can see from this table that the correlation, the Pearson correlation between the short chain fatty acid in the biofloc with Ankistrodesmus addition and the giant prawn was actually the lowest one compared to other treatments, indicating that the nutrient utilization in this particular treatment by the giant prawn was lower than the other treatment. So we suggested that this mainly because of 2 possibilities. The first is that giant prawn prefers to consume smaller bioflocs, or the second, the giant prawn can actually consume bioflocs, but they cannot digest or they cannot utilize the nutrients because of the incompatibility between the nutritional composition of bioflocs and the requirement of the giant prawn. So based on this study, we conclude that the biofloc nutritional quality and particle size is species specific. So based on what we have been doing in IPB University, we conclude that biofloc technology application allows intensive aquaculture system with efficient use of water and feed. And the quantity of bioflocs should be controlled at the maximum level depending on the system carrying capacity. This is mostly determined by the aeration capacity and mixing capacity. And also the culture species tolerance against high suspended solids concentration in the water. And based on our experiment, we can also conclude that microalgae can be used as a strategy to improve biofloc particle size and nutritional quality. However, biofloc nutritional quality or the utilization of biofloc nutrients can be species specific. And what is I would like to emphasize in this particular moment is that biofloc technology development still needs some improvement, and for this we will need multidisciplinary research and of course collaboration so that this technology can be ready as a simple technology at commercial level to support sustainable aquaculture. And thank you very much for your attention. This is from my department at IPB University, and for further info, you can try to Google Scholar to see if you have any interest in biofloc technology. Okay, thank you very much, Dr. Ating. The time is yours. Okay.
Thank you very much, Dr. Julie. It is very nice talk. And then we come into second Dr. Fajar, can you hear me?
Yeah. Yes, Dr. Ating.
Okay, thank you. Time is yours for 20 minutes.
Okay. Shall I share my PPT?
Oh, sudah kok. Sudah.
Okay. Okay. Good afternoon, participants. I would like to thanks to the Dean of Faculty of Fisheries and Marine Science and Head of Aquaculture Study Program and the organizing committee of this international webinar. Okay, this is the last presentation. So I will present my research about squid waste product for inducing shrimp immunity. Maybe this is the basic research, but it's inducing the friendly technology in aquaculture. Okay. As we know, the shrimp aquaculture is very popular in Indonesia, but with the increasing of intensive aquaculture, we have some problems in viral diseases. Many diseases attacks shrimp aquaculture, and we have to combat with this viral. So only two ways we can do it. We give antiviral, which we have found it yet now, and we increase the immunity of the shrimp. So it has to go to environmentally friendly and goes to sustainable aquaculture. Okay. What we have done in several years before, we did research about squid ink for shrimp with infectious myonecrosis virus or IMNV with symptoms that shrimp muscle and intestine necrosis from white and then become red and make mass mortality slowly but sure. And usually the shrimp farmer has to take out all the shrimp from the ponds. So we try to find the way to cut this viral attack. We try to use squid. We all know about squid. In Indonesia, especially in East Java, we have a high squid production with the ink, squid ink usually only have as a waste product. If we know the export commodity in fishery of Indonesia, we know high commodity from fisheries. for squid for export. So we have now a lot of squid production. So we try to use the squid ink as a medicine for IMNV. From LCMS tests, we use the squid powder. We found that the squid powder have betaine, cinnamic acid, and choline. Two of them is alkaloid, betaine and choline, and the others is cinnamic acid. From these ingredients, we try to make a prediction with in silico test. We know that choline and betaine can interact with ribosomal protein in virus. And then we add the squid ink powder in a shrimp feed. We use 3 treatments. And we can see that the squid ink powder can add the protein contents in the shrimp feed. And the result, after we infected the shrimp with IMNV, and we look at the muscle of the shrimp, we can see that giving 500 milligram per kilogram feed give a better condition for fat degeneration, necrosis, and vacuola in shrimp muscle. Can see here the result from giving the squid ink powder to the shrimp muscle. This is the best result is in 500 milligram of shrimp muscle, squid ink powder per kilogram shrimp feed. And then we look at hepatopancreas. It's also the best treatment for giving 500 milligram squid ink powder. You can see here hepatopancreas of the shrimp. And we look also at ribonucleic reductase, an enzyme in the muscle. In the left is negative and positive control, and the right side is treatment for 500 and 600 milligram squid ink powder per kilogram of shrimp feed. You can see the 500 milligram squid ink powder treatment give a different look. Not a lot of green color here. After we look at with CLSM, it means that 500 milligram squid ink powder give a better condition for the enzyme to attack the virus. And we also look at phagocytosis. We can see here that better phagocytosis. We can see the data after this, and also we look at superoxide dismutase, phenylphidase, and respiratory burst that all work in immunity system in shrimp. And from the data from non-specific immune response, we can see from total hemocyte count, and respiratory burst, SOD, phagocytic activity, and also survival rate. We look at the first week. We count it at week 0, and then we look again at the end of the experiment. At the 3rd week, we can see that the treatment with 500 milligram squid ink powder is better, all for THC, RB, SOD, phagocytic activity, and also survival rate. It gave 83% of survival rate. So from that result, we can see that total hemocyte count can increasing because the alkaloid compounds in squid ink, and this will increase immunity and recognition of foreign objects that enter the shrimp body. Also, the SOD, we know that the shrimp get infected with IMNV, it will produce reactive oxygen species, and it will balance with antioxidant enzymes, which is SOD, and this reduce the explosion of cellular superoxide during defense against IMNV infection. And this will protect shrimp cell from damage. So we can see the muscle of the shrimp. And from respiratory burst or RB, you can see the level of body defense of the shrimp. It's related to superoxide anion. It's characterized by the ability of blood cells to reduce NBT, and this RB value is come to phagocytic level. That the higher NB value is a better shrimp defense system in against IMNV. And also the PO, penile oxidase, we have the best result in 500 milligram squid ink powder per kilogram of shrimp feed. And we look also at CypA gain. This gain has important role in indicating the presence of pathogens in Vietnamese shrimp and fighting various stresses due to bad environmental condition. And also, usually we check it also for growth gain with other gains. So from this CypA gain, we know that lowest polymorphism value was obtained in treatment B, and the highest was in control. So it, from using squid ink powder, we know that this squid ink extract can increase non-specific immune system in Lithopanus vannamei against IMNV. And the last, we already used the squid ink powder for several experiments with fish, turtles, and so on. So we will do it later for shrimp production. So thank you for your attention. Time back to Dr. Ating. Thank you.
You can stop share, Dr. Fajar. Okay. Dr. Fajar, can you hear me?
Yeah.
You can stop share your screen.
Yeah. Sorry.
That's all right.
I'm looking for. Yeah. Okay.
Yeah. Thank you, Dr. Fajar. This very interesting talk about natural medicational treatment for shrimps. Because the presenter gave very interesting talk, therefore many questions for both speakers. Please share the questions. Okay. Many questions for Dr. Julie. Okay. The first one is from Prof. Pittman. Dr. Julie, can you hear me?
Yes, I can hear you.
Okay. You can read also. From Prof. Pittman about the specific effect of bioflocs. Do you think it's due to the effect on water microbiology interacting with shrimp, or it is because the shrimp can actually grow by eating certain microorganisms in the bioflocs? You can answer one by one.
Okay. Thank you very much, Dr. Ating. The first question from Professor Pittman. Thank you very much for the question about the species-specific effects. Species-specific effects of biofloc. Do you think? Okay, I think it's because every species of aquaculture organism has different or has a specific requirement for any nutrients, including amino acids, particularly amino acids. So that's why, based on what we found, we think that nutritional composition of bioflocs will also needs to be considered according to the requirement of the fish or the shrimp. We cannot, for example, consider that high protein content in a biofloc. For example, sometimes we can get like 40% protein in bioflocs. Is always good for every species that we culture, because apparently the protein quality is determined mostly by the essential amino acids composition, not only by the level of protein in a particular material. So I think we also need to consider more about the detail, more into detail into the nutrient composition. For example, as I mentioned before, the essential amino acids, because if this is not what the animals want, then the animals can consume the biofloc, but it's not necessarily that they can retain the nutrient coming from the bioflocs. It can be because the compatibility of nutritional composition is not the same as the required by the cultured organism, so that the shrimp or the fish just consume the bioflocs, but they cannot retain more nutrients from the biofloc itself, and it will of course affect the overall system performance. So this is still in question because we also we still have to do a lot of research to find what are the factors affecting the biofloc quality in terms of nutritional composition, because there are a lot of factors affecting this this aspect of bioflocs. For example, like microbial composition, the carbon source that we use, and as well as the environmental quality also has might also has a significant effects on the nutritional quality of bioflocs. I hope it can answer your question.
Any suggestion or
any comments?
Any comments? Professor Pittman, can you hear me? Okay, we go for the next. Okay. I had
muted myself and I had unfortunately turned off the camera. I think this is a very interesting approach, and if it's possible, I've told UNITA as well that I would like to cooperate with the other speakers here on seeing how we can develop shrimp-based health indicators on the gut and on the gills. And this would be a very interesting application of whether or not the gut is responding differently to different compositions of, for example, fatty acids and so on. Mm-hmm. We have already published articles on this in other species, for example, the sea bass and so on, but we haven't got it in shrimp yet. So this would be a very interesting thing to follow up, and we'd have to get some kind of international funding for this. But I agree with the approach. Very interesting.
Thank you, Professor Pittman. Yes, I'm looking forward to collaborating with you, of course. Okay, so shall I continue?
Next, yes, for the second question from Agus Sarif Hanafi.
Okay.
He also developing BFT applications.
Okay, thank you for the question, Mr. Hanafi. When I tried a Biofloc system into a particular species, what I usually try to identify first is to be sure that the fish is not sensitive to high suspended solids. Because if you have a fish species that is sensitive to high suspended solids, then applying Biofloc system in this species, especially if we culture the Biofloc or we generate the Biofloc inside the culture system, then we will have problems. So first, you have to make sure that the fish can stand with the high suspended solids. Otherwise, we can still apply Biofloc system, but we culture we have another setting, another tank, for example, for Bioflocs that is separated from the culture tank, so that Bioflocs mainly function as a biofilter. That is another strategy. So that is the first thing that I will do if I want to apply Bioflocs in specific species. And then secondly, we have to make sure, or we can also find out by research, is that the fish or the shrimp can consume Bioflocs. Because otherwise, if they cannot consume Bioflocs, then there will be an accumulation of biomass in the system that will require some maintenance. We can still do that, but again, an ideal Biofloc system cannot be attained if the fish or the shrimp cannot consume Bioflocs in considerable amount. So you can use that approach. So for example, for Pangasius and Ostronemus gurami, we have already tried, and they can grow quite good in Biofloc system. As for Halostoma, Trichogaster, Chana, and Anabas, we don't have any information, but maybe if you have already tried, you can share it with me. I hope Mr. Hanafi, I can answer the question of Mr. Hanafi. Mr. Hanafi, can you hear us?
Thank you, your Sajus Ekasari.
Okay.
That's enough.
Okay, thank you very much, Pak Hanafi.
Thank you.
And then the next question, did you experience or encounter mass mortality in cultured species using... Okay. Yes, I have an experience with mass mortality when I first tried Biofloc system in African catfish. How did I handle it? The mass mortality was occurred in the first few days of culture. So we thought that this mostly because of the quality of the fish. The fish juveniles, which was not good. So it cannot stand with the new environment, especially in Biofloc system. So that as soon as we transferred the fish into the Biofloc system, most of the fish died. But when we have good quality juveniles using the same species, the system works perfectly fine, and we can harvest the fish into consumption size. So again, I think the juvenile quality plays also an important role in the success of Biofloc system. Handling techniques and measures to prevent the situation. Okay, so to anticipate this problem, you will need to be sure that you have a good quality fish juvenile or good quality seeds that is healthy and also uniform in size and free of disease. I think that is the common that's common in any culture system, yeah. But in Biofloc system, it's also very important. Can you give us an alternative treatment that is locally available to lower the ammonia content in Biofloc system? I'm not sure I understand what the question is. Because usually in Biofloc system, we can maintain ammonia concentration at a low level. So if you have a Biofloc system with high ammonia, it means that your system is not good enough. Whether most of the time it's because of the CN ratio, which is lower than 10. Or it can also be because of the failure in your Biofloc system. For example, if you have problem in aeration, then the heterotrophic bacteria cannot work properly. Then you will have some mortality in the bacteria that can also contribute to the increase of ammonia level in your system. I think that will be all. Dr.
Still two questions for you, Dr. Julie,
okay.
Because many people want to meet you actually. For the next questions, please.
Yeah, okay. Dr. Azman, thank you for the question. Well, for the Biofloc volume, it will depend on the culture species and also the carrying capacity. For example, for tilapia, they can withstand with high floc volume. I have an experience with floc volume of more than 100 milliliters per liters, and the fish can still grow. But the problem is the water's quality become unstable. And especially when you have especially when the aeration system is not so good, then you will have, because high biofloc density means high requirement for oxygen, means that we will need to have a backup system for the aeration. And also when something happens, and also when the temperature goes high, it will increase the requirement for oxygen. And because we are living in tropical area, sometimes, most of the time, during in the afternoon, there's a significant increase in temperature. Then it will also increase the metabolic rate of the bacteria, and this, of course, will increase the requirement for oxygen. So I would suggest for tilapia to control your floc volume not more than 100 milliliters per liter. Then you will have a safe room for any to anticipate any problem with aeration or electricity or other things. As for shrimp, I think the volume of biofloc should be maintained at a lower level, even lower than 50 or 20 milliliter per liter, because otherwise, again, you will have the same problem in oxygen consumption. And I think for shrimp, they are more sensitive to oxygen concentration. Okay.
Thank you, Dr. Julie. Oh, you can continue.
Oh, okay. Okay. Thank you, Professor Maktu, for the questions. Could you please inform me how many species of microbes in your biofloc? Okay. In my opinion, well, first of all, we have never, based on our experience, we rarely measure the density of the of the bacteria. Why? Because, of course, in a biofloc system, the density of bacteria is always high. And in order to determine the species of the bacteria, I think the common method right now is by plating plate count, which is, for me, it can be doubtful, because the one that can grow in plate condition, in agar plates, might not be the one that is dominating the culture. And I think for this, we will need something like, for example, more advanced tools like next generation sequencing or other tools which can identify the dominant bacteria in real time. So not based on what we can culture, because what we can culture is not necessarily the same as what dominates the system. That is in my opinion, but I might be wrong. And then also the microalgae in my biofloc. Okay, for the microalgae in my in the biofloc system that I presented before, I use chlorella and Ankistrodesmus. Chlorella is the easiest one, because it's relatively easy to culture, and we can obtain quite high density in a short period. I think that would be all. Yeah, Dr. Ating, I hope I can answer most of your question, but if not, you can contact me by email or by WhatsApp.
Okay.
I think the organizing committee has my number and my email address. Thank you very much.
Thank you, Dr. Julie. The next questions from for Dr. Fajar. Dr. Fajar.
Excuse me, Pating.
Yes.
Oh, yes. Prof. Maktu.
Second question is not answered.
The second answer?
My question is not answered by Miss Julie.
Okay, the second answer is amino acid in your biofloc must contain microbia and algae or produced by Biofloc. Okay, because the time is very limited, we can send our question to Dr. Julie, and Dr. Julie can answer through email.
Yeah, I will answer. I will answer by chat.
Okay. Because the time is very limited.
Okay. Yeah.
Okay. The question for Dr. Fajar, please. Okay. The first question is, what is the optimum concentration that you suggest to improve productions?
Okay, I will answer it, Dr. Ating. Thank you for your question, Reski Dona Maliki. The first question, what is the optimal concentration that is to improve production? I give 500 milligram of squid ink powder per kilogram feed. That's for big shrimp, for juvenile till big shrimp. And the second question, is your product economically viable when we apply it in the shrimp culture? Yes, I have tried to make some squid ink powder production, and I count about the economic value. And this product is economically viable for the shrimp culture. You can read in my article in journal about squid ink, and you can make some calculation from that. How about that, Rizky? It's okay?
I think it's okay. Okay, for the next question.
Okay, the next question, how to produce your product in commercial scale? We need, from Dani Arianto, of course, we need a lot of squid ink, but this is a waste product from squid. Usually we didn't use the ink, so you can make this product squid ink powder. Of course, for commercial scale, we use some equipment, big equipment for that. So we can produce it in a commercial scale. Of course, we need a lot of squid ink ingredients. But I have tried, and we still work for that, for commercial scale. Okay, and the third questions, is the squid extract can be applied to the fish or shrimp larvae? Yes, of course, we can use it for immunobooster. We usually for larvae, I've tried with dipping technique, and for immunobooster, usually the immunity will increase after 2 weeks, so we can make it as a booster after that every 2 weeks. I think like that, Dr. Yunita.
Okay, thank you, Dr. Fajar. Okay. Because time is limited, to sum up these sessions, we realized that Biofloc is eco-friendly fish farming technology Biofloc technology offer benefit in improving aquaculture productions that could contribute to the achievement of sustainable development goals. This technology can be an innovative strategy for minimizing waste, efficiently use of feed nutrient, good reproductive performance, and for disease control and prevention, and also could result in high productivity. Development of natural medicational treatment and immunostimulant, such as from squid waste product, is an alternative for disease control toward sustainable aquaculture. Thank you very much. We come into the end of these sessions. Thank you very much for our both speakers, for her and his valuable contributions and sharing their knowledge with us. I hope our collaboration will continue in the future. Ladies and gentlemen, may I thank you very much for your active participation in this webinar. I hope you enjoy this second session of webinar. I pass on the control to Master of Ceremony. Thank you very much.
Thank you very much, Dr. Ating, Dr. Julie, and Dr. Fajar, and also all the participants that are very patient until now. I would like to request to you all to turn on your camera once again to have a photo session, because right now we have increasing participant. So, are you ready? Okay. Let me count. There are 3 phases. 1, 2, 3. Okay, next phase. 1, 2, 3. Okay, next one. The last one. 1, 2, and 3. Yeah, thank you very much, everyone. So, ladies and gentlemen, we come to the end of the event. On behalf of the Committee and Fisheries and Marine Science Faculty, Universitas Brawijaya, I would like to give my great gratitude to our remarkable invited speakers, and I would like to, and also to entire participants who joined our webinar from the beginning to the end. And also, I would like to apologize if there is something that unpleasant happened during our event. Kindly find the attendance form so we can send the e-certificate to your email, and the e-certificate will be sent within 1 week from today. Please be patient. Once again, thank you very much. We'll see you on the next webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you.
Thank you.